My name is Russ Altman. I'm on the faculty at Stanford in a number of departments, including bioengineering and genetics. They pay me. And then I have an appointment in medicine and in computer science, and I live nearby at Stanford from New York originally.
That's great. And you are one of the creators, the creator, I guess, of PharmGKB?
It's a team. I wrote the original proposal and have continued to be the principal investigator. It's now in its 13th year. We started back in 2000.
Great. And so for somebody who's just seen PharmGKB for the first time, what is it all about?
So we are totally excited about how genetics can influence your drug response. And back in 2000, the NIH and others realized that there needed to be a central repository to gather all that information together. So the mission of PharmGKB, simply put, is to understand all human genetic variation that impacts drug response, and to catalog it to make it available to researchers and eventually practitioners so that they can use it in their work. And it's a one-stop shop for the genetic influences on drug response.
That sounds like a mountain of information.
We do this in two ways. We have human curators who are experts in pharmacogenomics, and they read the literature literally every week as the journals come out. They scan them looking for genes and drugs and their relationships being discussed, and then put them on a queue for human curation. This has become difficult, and so the second effort in the lab are software tools to do this. So we do a lot of natural language processing, data mining, and stuff like that to try to help put good articles in front of the curators so that they can be more efficient. We do still want humans in the loop because this is sufficiently tricky, and the end goal being it might actually affect patients. So we're not ready to close the loop and just have computers doing it all. So there's always a human in the loop so far.
Well, who's the intended audience for this? Is this sort of opened up mainly to clinicians or science people or anyone?
It's a great question, and that has evolved. For the first 5-7 years of the database, it was specifically just for pharmacogenetic researchers, to let them know what was known and what was not known, really so that they could pick the problems to work on and not reinvent a wheel or be unaware of some important discoveries that have been made. With the success of the field, however, it has become increasingly apparent that some of this stuff is ready to roll out in clinical practice. And so we are now involved in consortia where we're actually writing clinical practice guidelines for clinicians to actually use pharmacogenomics in practice. And that's probably where we'll stop. We'll probably go all the way from the raw data all the way up to the clinical advice that you would give to a physician who's making a decision about drug prescribing.
Wow.
And that's really exciting because that's how you actually impact patients, and that's why we do this. We did this because we all believed that the genome was going to be important for health, and your response to drugs turns out to be one of the key early uses of the genome that we'll be able to benefit patients with.
So if you were an individual, just a layperson who had personal genetic information about, let's say, a gene that affects the metabolism of a certain drug, you could take your variation and go to PharmGKB, and what would you find out?
So at PharmGKB, you would find a couple of things. You would find the entire literature on that drug-gene pair indexed as to how good was this, not how good was the study, but how big was the study, how compelling were the statistics when they did the analysis, and you'll find all the individual papers. But what you'll also find is products that our team has created to kind of integrate that data. So these clinical guidelines are one thing that you may find. These require a lot of evidence, so there's not as many of them as there are things for weaker evidence. There's about 12 of them where we can really put down clinical advice. But our curators also put together pathways of drug response to show what's happening in the liver or what's happening in the effector cells that are affected by all of these genetic variations. They also write summaries. This gene is important because, and they actually publish these summaries so that somebody who wants to learn about a particularly important gene. So a patient, unless they have scientific background, would not find too much. A physician would find those clinical guidelines and the supporting evidence, and a researcher would find, well, a candy land of the raw published literature, and then what we hope are very useful summaries of it to help them think about what the next research questions should be.
Yeah, it sounds like a great amalgamation of the information that exists, but in a centralized place.
We call it a knowledge pyramid. In fact, if you look at the site, pharmgkb.org, please visit it. PharmGKB.org has this knowledge pyramid where we have the raw data that's published in the literature, and then we have aggregations of it, and then we have filtering of it, and then we have the clinical advice that we give to doctors. And that's really how it works. You have all this research that eventually impacts patients.
Do you keep track of how many entries you have? Like how many drug genetic variation responses?
Absolutely. So I do annual reports for the NIH.
Okay.
And so over the years, we have totally instrumented the site. We know how many hits every page gets.
Wow.
We know the most popular pathways that people look at, the most popular genes.
Name one.
So people love codeine. And people, it's
Don't
a pain,
they?
it's a
Don't
pain,
they?
Exactly, it's a pain reliever. People are very interested in warfarin. For the pathways, those are the ones they like. For the genes, they like some of the genes that are involved. There's a gene called CYP2D6, terrible name, cytochrome P450 2D6, which is very important for hundreds of drugs, and that's one of our top hits.
Wow. And what are people using when you say it's a top hit? These are probably researchers using this information?
Yeah. So we learned a long time ago that people like the Google type interface. They don't really want to do super duper searches with lots of bells and whistles. They want to type free text. So we've optimized our free text search on the site. The nice thing about that is it's really easy to look at the free text searches and to categorize what's the average length, how many words are included, and what genes, drugs, and diseases are being mentioned. And we can then use that kind of to do market research for the site to improve it, because we see what people are typing in, and we can type those same things in and say, do we like the answers that we're giving when somebody types that in?
So what would you say what kinds of tools would you like to see on the PharmGKB site that maybe aren't there now?
Great question, and we're thinking about this rapidly. This has been a website for 13 years, but guess what? There's these things called mobile devices. We haven't engineered the site for that, but it is loud and clear from our users that they are at conferences and somebody mentions a gene and they're going to go onto their smartphone or their iPad or their Galaxy, whatever, and they're going to type it in. And so we're working very hard at how do you take all this information and just give the highlights in a much more restricted real estate stuff.
Sure.
People all over the world are worrying about this, and we are too. So there's that. But it's funny, the other thing is we have a very textual-based site, and it's good if you want to read text, but it's come to our attention that people, even when they have a lot of monitor space, they want to look at prettier graphics. They want to see networks versus text. So we're also thinking of refactoring the whole site to produce a network way of thinking about pharmacogenomics. Like, here's a drug and here's all the genes that connect to it, or here's a gene and its drugs, or here's a network that connects genes and drugs. And so we're also prototyping that kind of stuff. So that's kind of user interface type things that we're doing. On the content side, we're very interested in pushing these natural language processing because our curators are starting to see that they work very hard and the literature is starting to get them overwhelmed.
What other resources would you recommend? I mean, do you want PharmGKB to be sort of the one-stop shop, or are there other useful resources that you would recommend to people sort of in conjunction with this?
Absolutely. So a couple things. We have this tab-based display format right now, and one of the tabs is other resources. And it's very important because what we can do is, since we know the drug or the gene that the user is currently looking at, we can have super specific links to other information sources about those drugs, about those genes, about the diseases that they treat. And so we have those links, and we are always looking for more links because this is the beauty of the World Wide Web, is you can make these connections. In addition to that, sometimes a data source is so valuable that we, with the permission of the people who are creating it, bring it into the PharmGKB so we can more tightly integrate that data with our data. So there's kind of two phases of the relationship. The we'll definitely link to you phase, and then the we love you so much that if it's okay with you, we'd like to bring you in and make you part of our interface, part of the way we represent information. And these include basic biological databases like GenBank, which holds DNA sequence. There's a very valuable drug resource called DrugBank, which we link to. There are the dbGaP, which holds GWAS studies. And a long list of other links that you would find on this site.
I'm curious, is online Mendelian inheritance in man part of that?
OMIM, or online inheritance, great resource. Kills us in some ways because it's great, but it's text-based.
Yes.
And it's not super structured.
No.
It's hard to write computer programs to parse it. And so, but we do that because it's incredibly valuable, yeah.
You mentioned that one of the most popular hits of things that people are interested in are the warfarin pathways.
Yes.
Now, why is that?
So warfarin is pretty much the most advanced case of pharmacogenetics that has been studied. Why do I say that? Warfarin has more than one drug that impacts its response, and we actually know what a lot of those, more than one gene, I'm sorry. Warfarin has more than one gene that's important for understanding its response, and we actually know what a lot of those genes are and how to combine the information from more than one gene. That's a frontier. There are many drugs for which we know there are several genes, but we don't know how to combine the information yet. We do for warfarin, so that's the first thing. The second thing about warfarin is that we also know some of the environmental variables that affect warfarin response. How much green leafy vegetables you're eating, what your height and weight is, what other drugs you're on. That all combines to have warfarin be the single best drug that we have for predicting response based on a combination of genetics and environmental factors. The other thing that I should have actually mentioned right off the bat is there are something between 30 and 50 million Americans alone on this drug, and probably, obviously, twice that or more internationally. So we're talking about 100 million people on this drug. There are new drugs coming out that are hoping to knock it off as the leader, but today, warfarin is still an incredibly important drug worldwide, and so accurate dosing, especially for a dangerous drug like warfarin. As you may know, warfarin is an anticoagulant, and it makes it harder to clot your blood. That's good if you've had a heart attack. It's good if you've had a stroke. It's good if you've had a clot in your lung or in your legs. But if you have too much of it, it can kill you with internal spontaneous hemorrhage. So there's this very narrow range we have to get it right, and that's where the genetics can help.
Right. It sounds like blood clotting is obviously important when we cut ourselves and we're bleeding, but if it's blocking our hearts, that's not so good. Well, you mentioned that there's so many people on warfarin.
Yeah.
The variation in the human genome that exists, on the PharmGKB site, for example, does it sort of separate? Is it able to document the pharmacokinetic and the pharmacodynamic pathways separately? Does it combine them? How do you present that information?
Yeah, so those are two separate. Good for you for knowing about this, and good for your class. In the case of warfarin, there are both pharmacokinetic and pharmacodeterminants to the response. And there are genetic variations on the pharmacokinetic side that change the rate at which it's metabolized. And one of the most important genes is called CYP2C9. There's another one called CYP4F2, but 2C9 to take as an example. It's well known that if you're going to metabolize warfarin more slowly, you're going to need less of it. And if you take the normal dose when you're a slow metabolizer, you're going to wind up with effectively an overdose and too much bleeding. There, on the pharmacodynamic side, there's a gene, again with a terrible name. It's called VKORC1, or vitamin K oxidoreductase component 1, which is actually the target. It's the protein which the drug warfarin binds to inhibit clotting. And there are some variations both inside this gene, but also in the control regions of the gene that control its expression, that markedly change its levels. And there are some variations that are very important for determining a warfarin dose in that. So it's a great story because there's this one PD gene that seems dominant, this one PK gene that seems dominant. Together, you can come darn close to predicting what the right dose will be for an individual patient.
Even without needing to see them clinically first.
That's right. You do best if you have their age, because there are some age effects, and gender. There's some sex effects, the genetics, and then ethnicity. You would hope that ethnicity would drop out, because you would hope that once we have the genetic measurements, who cares what your ancestry is? But I must say that our ability to predict dose for African descent people is still not as good. And so in our lab, we're working very hard. The hypothesis would be that there are additional genetic variations seen in that population that have not been studied yet, that with knowledge of those, we'll be able to get our level of prediction for African descent people to be as good as where we are now for European and Asian descent.
That's wonderful. I want to ask you one more question generically. If somebody's taking this course and they are super excited about the idea of pharmacogenetics, pharmacogenomics, what advice would you give them to sort of pursue this interest?
So as a patient or as a researcher?
A learner.
As
both.
a learner.
Do both separately.
Well, so as a patient, there are increasingly companies out there that are not only providing, so I know that your class knows about 23andMe. That's not meant for clinical purposes, but nonetheless, the quality is quite good, and you could get at least a suggestion of what some of your drug responses might be. And now there are more formal, clinically approved testing facilities that will check your genetics and then give a report to your physician that would allow them to, your physician, to make decisions about drug dosing. In terms of learning more, there are some conferences. I'm the president of the American Society for Clinical Pharmacology, which has an annual conference where pharmacogenomics is featured. It's not the only thing, but it's one of the things at this conference. The American Society for Human Genetics has a great conference. It's going to be in Boston in 2013. That makes this tape dated, sorry. It was in Boston in 2013. There are also actually, there's Twitter feeds, and there's something called the Genetics Alliance, which is a bunch of a patient grassroots group that wants to see more genetics used in medicine. My friend Eric Topol in San Diego has a great course on genomics in medicine that he teaches down in San Diego every year for clinicians, and I bet you there are some non-clinicians who attend that. And then there are conferences that kind of pop up all the time. So I think that's one way to do it. It's getting increasingly played in the media because it resonates with people very much that their genetic background would affect how they respond to drugs. So it doesn't take long to convince somebody that this is not an unreasonable thing to think about. And unfortunately, we don't know how our parents. One of the things that we don't get from grandma and grandpa is how they've responded to their drugs in general. And you inherit that just as you inherit your height and your eye color, but that is often lost in the family lore, and so genetics now gives us a chance to recapture that so we can use it for the patient's benefit.
So tell me, way of the future in medicine or not?
Absolutely. So I would be disappointed if in the next 10 years, I'm already aware of local implementations of pharmacogenetics in some of the leading places, St. Jude's Medical Center, Vanderbilt, University of Maryland. We're trying to get things going here at Stanford. So some of these places are already giving trials. I think those trials will be successful, and then you'll start seeing it becoming kind of a standard of care. And at that point, there'll be an expectation that this is part of the armamentarium in medicine, which will be incredibly exciting. And then all of this work we've been doing, putting together the database, writing the guidelines, will kind of have its desired ultimate effect.
Thank you so much for chatting with us.
It's my pleasure. Thanks for doing this.
Yeah, of course.